Okay, so let's get into this because today we are doing a deep dive into the grim reality of glioblastoma, GBM.
Yeah, for decades, this has been one of the absolute deadliest adult brain cancers.
And the prognosis has just been heartbreakingly stagnant. I mean, we're talking a median survival of just 12 to 15 months, and that number hasn't really budged since 2005.
It's hard to overstate how stuck the field has been. It's what you call a triple failure cancer.
Triple failure.
You fail to deliver drugs past the blood-brain barrier, you fail to get the immune system involved, and recurrence is, well, it's pretty much guaranteed.
But that's why we're here. This new research we're looking at from Washington and Northwestern, it seems to crack all three of those problems at once.
And it does it with something as simple as nasal drops, delivering these things called spherical nucleic acids, or SNAs.
It sounds almost too simple. But before we get to the solution, let's just quickly set the stage. Why is GBM such a fortress?
Well, the first barrier is the obvious one, the BBB, the blood-brain barrier. It's designed to protect your brain, but it's almost too good at its job. It blocks something like 98% of small molecule drugs.
So you just can't get the medicine to the tumor.
Not in a high enough dose, no. And then there's the cancer itself. It has this highly infiltrative phenotype.
Meaning it spreads out.
Exactly. Think of it like a plant with microscopic roots spreading deep into the healthy brain tissue, far from the main tumor mass. A surgeon can cut out the bulk of it, but those roots always remain.
And the immune system, which should be the cleanup crew for that microscopic disease, it just doesn't see it. The source is called the tumor cold.
Totally cold.
Mm-hmm.
GBM is a master of disguise. It surrounds itself with these immunosuppressive cells, specifically M2 like macrophages. They basically send out a signal telling the killer T cells to stand down.
So a checkpoint inhibitor, the standard immunotherapy, would be useless.
Completely. You're trying to hit the gas on an immune system that's been actively sabotaged. There's no one there to do the work.
So that brings us to the breakthrough. The vehicle they use to solve this is the SNA. What's the secret to its architecture?
It's just so clever. It's a tiny gold nanoparticle, maybe 13 nanometers, and it's covered, densely covered, in custom DNA strands.
And that's the SNA effect they talk about.
That's the one. That structure lets it slip into almost any cell without needing toxic agents, and maybe more importantly, it protects its DNA payload from getting degraded by enzymes in the body.
Okay, so that's the vehicle. But how do they get it past the blood-brain barrier? That's always been the core problem.
Right, and this is the absolute game changer. They don't. They bypass it completely.
They bypass it. How?
With the intranasal drops. The SNAs don't just seep in. They actually travel along the trigeminal nerve.
Wait, along the nerve itself?
Yes, it's a direct highway into the brain. They use what's called perineural and extracellular transport to go straight into the brainstem and parenchyma. It's rapid, too.
Well,
They detect it in the brain within 4 hours.
that's astonishing. And because it's not going through the bloodstream, you avoid that systemic toxicity, that cytokine storm that can be so dangerous.
Exactly. The therapy stays where you want it, in the brain.
So once the SNA gets into the tumor cell, it releases its payload, this DNA sequence called ISD45. What does that do?
The ISD45 is engineered to look just like a viral invader. It's a powerful alarm signal for the cell's innate immune system, specifically for a pathway called CGAS sting,
So it's basically like setting off a burglar alarm inside the cancer cell.
a very, very loud one. Activating sting makes the cell pump out type I interferons and a ton of chemokines. And that does two critical things.
It turns the tumor hot.
That's the goal. First, it forces those suppressive M2 macrophages, the tumor's bodyguards, to switch sides. They get re-educated into pro-inflammatory M1 cells that attack the cancer.
And the second thing,
The chemokines act like homing beacons. They send out a powerful signal that recruits fresh cytotoxic T cells from all over, drawing them right into the core of the tumor.
and the results in the preclinical models were stunning. Eradication of established tumors, plus long-term immunological memory. It's like an in situ vaccine.
It is, and they found this really intriguing detail, a sex dimorphic response. The therapy works significantly better in female mice,
Which is a huge clue for future human trials.
a massive one. It tells
Yeah.
us that our innate immune systems might be wired a little differently, and we have to account for that.
So the big takeaway here is this could shift GBM from, frankly, a palliative death sentence to a disease with real curative intent. And the delivery method, a simple nasal drop, is just transformative for quality of life.
You're creating a new highway into the brain. And think beyond cancer. If you can deliver this, maybe you could deliver neurotrophic factors for Alzheimer's or gene editing tools for Huntington's.
The discovery of the trigeminal nerve as this fast, targeted route is just monumental. Which leads us to a final thought for you to chew on. If we can bypass the blood-brain barrier this easily, this non-invasively, just by targeting a specific cranial nerve, how many other intractable brain diseases could be solved with something as simple as a spray?